So let me start with a story. So about 15 years ago, I found myself on an airplane on my way to Washington, D.C. to testify in a Senate subcommittee hearing. It was a subcommittee chaired by then Senator Arlen Specter, who is, if any of you knew him, quite the bulldog about extracting information from whoever he's cross-examining. Specter was previously a prosecutor. And the hearing that I was sent to testify at was a hearing about human embryonic stem cells, which had just been isolated for the first time in Jamie Thompson's lab. And I was testifying in that hearing along with a young woman who had ALS, Lou Gehrig's disease, and then also a fellow named Christopher Reeve, who some of you have probably heard of. Needless to say, this was a media circus. And the purpose of the hearing was to describe, in terms that even a senator could understand, why stem cells were so important to the future of medicine. Now, I could not compare to Chris Reeve's star power, but what happened was that really began what has been a 15-year journey from me, from a scientist who used embryonic stem cells in the mouse to analyze basic biological pathways, to somebody who's now trying to use stem cell technology to develop a better understanding of human disease and to develop novel treatment approaches to human disease. What you might think about is, what are the best ways to think about this technology? And again, I go back to a story. So how many of you saw the movie The Graduate with Dustin Hoffman? Yeah, pretty much everybody who's an American, and many, many non. And you'll recall that this movie had a couple of major points. So one major point was older women can be interesting. That was pretty good, right? I'm actually married to an older woman since I am an older man. And second, do you guys remember the career advice that Dustin Hoffman received as a young college graduate, wondering what he was going to do with the rest of his life? Plastics, right? Everybody remember plastics? Okay, so why was that? So if you think about it, in the 50s and 60s, most manufacturing was based on natural materials. Wood, metal, and that was about it. Your cars actually were constructed largely out of metal and then rubber tires. If you fast forward now to 2014, if you look around, what you'll see is just about everything we use, including electronic devices, are largely constructed out of plastics, right? And that's because you can make plastic from simple materials, basically oil, and plastic has this amazing ability that allows you to shape it into almost anything with all sorts of different tensile properties, melting properties, what have you. It was great career advice in the 60s. Now, if I were remaking that movie today, which I'm not going to do, I don't have the time for it, but if I were, I would change the advice. And the advice would be stem cells. And the reason for that is that stem cells are, in fact, the biological plastics of this century. So what do I mean by that? So imagine, and this is actually true, but imagine that you have a biological material that you can make more of by feeding it. This is good. We know how to feed things. We can grow crops and animals. So you feed them so you can make more of these cells, which are the basic unit of how we build organisms. And then imagine that you've learned how to tell those cells how to make every adult cell type that we find in a human adult cell. Nerve cells and heart cells and skin cells and blood cells. Imagine that you can do that in the lab and that you can eventually turn that into manufacturing. What that gives you is what I refer to as biological plastic. I sometimes refer to this building to lay groups as the plastic factory. And the reason is that is in fact what stem cells allow you to do. Now we're just learning to have basic control of those processes, but this is effectively what is going on in this field at large. Now, you might ask, who cares if you can make large numbers of cells of a defined type. And of course, one answer to that is if you think about many diseases that people have that show up to our clinics, a lot of those diseases are diseases where cells have died and are gone. So for example, type 1 diabetes, juvenile diabetes, is a disorder where the beta cells of the pancreas are dead because the immune system attacked them, and as a result, kids or adults who don't have beta cells cannot make insulin, which is the normal role of the beta cells. And so you can see that if you had a way of giving people new beta cells, this could in principle treat their diabetes. Or imagine that you have a heart failure patient where a large number of cells in the heart have died. If you could figure out a way to make heart cells in the lab and then put them back into the heart as a patch for the scar tissue or what have you that's been disrupted in the course of a heart attack, then you could treat people who have heart disease. And that's actually a very important potential use that we and others are trying to bring from the blackboard to our clinics over the course of the next several years. What you might think of as simple cell replacement therapy, making new cells to replace cells that have died in disease. So that's one really important and obvious use of the technology, and it's one use where this ability to grow large amounts of cells and tell them what to do becomes very important. Now, note, we haven't figured out all the rules yet, so we can't yet do it with every cell type, but at some level this is now a problem of development, figuring out how to make each and every type of cell that has perished in one disease or another. Now, it turns out there's a second use of this technology that is as important or perhaps more important than the one I just described, which is cell replacement, and that is using stem cell technology to figure out what goes wrong in disease, okay? Now, why is that so important? You probably know that it costs about $2 billion is the current estimate to develop a new successful drug. Now, why is that? It's not just that CEOs of drug companies are flying around in private planes. That costs a lot more, or planes cost a lot less than $2 billion per therapy. The answer is that most drug development efforts fail. In fact, the current number is that 90% of drugs that start clinical trials don't make it through to the end. And so the failure rate is 90%. That's terrible. The reason the failure rate is so terrible is not that pharmaceutical companies are stupid. Actually, they're not. A lot of really smart people work in these companies. The problem is we don't actually understand very well what goes wrong in most diseases, and in part that's because we don't understand actually how the normal situation works very well. Our understanding of the biology of cells is very limited, actually, relative to what we need to know. And so when cells are malfunctioning or dying in disease, the problem we have is we don't understand how it should work normally, and therefore we don't understand what kind of therapy we're trying to build to make a successful drug. Now, you probably know that at present, what we do is we use so-called animal models of disease, which are really important and very valuable for understanding the principles of what go wrong in different sorts of diseases and how cells normally work. But I'm going to bet that you've noticed something about humans, and that is we don't look like big mice, right? We're actually a very distinct species, and in fact, the biochemistry in our cells is actually very different in many cases from what works normally in mouse cells or dog cells or cat cells or what have you. And so at the end of the day, one of the ideas that we're putting into practice here is that if you could make human cells in the lab in a dish that actually had the disease that you're trying to treat, you could actually use this as a way of figuring out what's really going wrong in that disease and as a way of testing drugs on actual human cells before you even get to human clinical trials. And so, for example, my lab, which is in this building, folks in my lab have developed versions of human Alzheimer's disease with human neurons, human brain cells in a dish. We do this on a daily basis. And that's important because mice don't actually develop Alzheimer's disease. It's a very human-specific kind of disorder. And we're now able to use this technology to start understanding the pathways that go wrong, for example, in Alzheimer's disease. And we have an effort with folks at Stanford Burnham to test different drugs that have already been approved by the FDA to see whether they'll fix the defects that develop in these Alzheimer neurons. So that's a second use of this technology, which is actually very important, that will let us understand how human disease actually works in true human cells. And what I've argued over the past years is if you could change the failure rate from 9 in 10 to 8 in 10, you completely revolutionize the industry because you've doubled the success rate. And I think that's something that's actually within range. But we'll see. That's one of the big experiments. Now, the final use of this technology, and then I'll close and let you folks ask questions, is what you might call put amazing tools in the hands of really smart people. And what I mean by that is, if I can imagine a use for this technology, it's already boring, right? What we really want is to find really smart, creative people who are going to get their hands on this technology and come up with things we haven't thought of. And I'll just give you a good example of past history where that happened. So in the 70s, when I was a graduate student, recombinant DNA, gene splicing technology, was just being developed. And there were many ethical and scientific concerns about putting human genes in bacteria, and maybe we'd make the bacterium that ate Harvard and things like this. And there was a lot of fear and loathing and all the rest. But one of the arguments that kept things moving ahead was the statement that we were pretty sure that you would be able to make human insulin in bacteria someday, and that would be really good for diabetics, who at the time were limited to pig insulin, and not all of them could tolerate that. None of us realized at the time the degree to which recombinant DNA technology was going to revolutionize many different fields. And in fact, if you fast forward to where we are today, you can now get human insulin and many other human proteins that are made in bacteria and that are therapeutically very valuable. But if you also ask the question, were there any unanticipated uses of recombinant DNA technology? The answer is you bet. And the simplest one to remember is that this technology found its way into the courtroom, where the use of DNA-based technologies developed as a result of moving ahead aggressively with that kind of research is now used to declare who was at the scene of the crime, who held the gun, whose blood is that, all that sort of stuff, whose hair is here. It was a completely unanticipated use of technology that developed because smart people got their hands on novel tools, what you might call enabling technology, which, as you know, means technologies that have broad utility. And this kind of stem cell biological plastic technology is exactly that kind of technology. Okay, so that's all I want to do in terms of prepared remarks, semi-prepared. Why don't we go ahead and let you folks ask questions to drive the conversation? Yeah, thank you.